that maybe has something to do with the maintenance. But bottom line is no one knows exactly what any of these do, and all of this is sort of still testing stage. This is a diagram that just sort of shows you the same thing. This is the same protein here. Right? That's ATP. Here's the A beta portion. Remember that beta amyloid peptide is about 40 to 42 amino acids, and that's the blue region here. This is the AP protein. There's a single helix that goes through the membrane. One thing that we sort of suggest something about this thing is that it is modified. It's sulfated. This region here is called API, which stands for Punnett protease inhibitor. And there is a protein that inhibits proteases on the outsides of cells. And this sequence here is homologous to it. So we think that sequence protects this thing as it sticks on the outside of the cell from being digested by proteases other than that secretase. This shows you where the beta, the alpha, and the gamma secretase is cut. Now, if this alpha secretase cuts and one of these gamma secretases cut, the fragment that's released is about 26 or so residues, 1 to 28. So 1 to 40 is 26, and that's 28 amino acids. So from here to here. And so if you have a cut here and a cut here, you make this small peptide, 1 to 28, and our cells are able to handle that. And they take this amyloid peptide that has 28 amino acids and break it down further with proteases to smaller pieces. If you make a cut from beta to one of this gamma over here, then you generate something that has 42 residues, 1 to 42 residues. From that aspartic acid down to this alanine residue. That's that lysine. Oh, anyway, this guy here is from here to here, 44 residues. I'm not sure why I put that other thing up there. So the processing is done by these enzymes called secretase. Here's the protein that's in the membrane here. Right? Here's ATP. And there are three enzymes that process it. One is called the gamma secretase. Here's the gamma secretase. And it's rather unusual protease, right? What is that protease doing there that's sort of unusual? Anybody have an idea? Should we talk about proteases at all in biochem? Well, most of the proteases are aqueous proteases. They work in the aqueous environment. But this particular protease, this gamma secretase, right, is cleaved inside the membrane in the lipid bilayer. And that's kind of unusual. There are other examples, and I'll show you one in just a minute, but that's sort of an unusual cut. This gamma secretase, right, which is the first of these secretases that was discovered, is called presenilin, because it essentially is presenilin because it leads to senility if there's mutations in it. Gamma secretase was actually discovered by a guy named Samson Sodia, who is now the director of the Molecular Biology Institute at the University of Chicago. But he was a graduate student in biochemistry, so in our department here. And he discovered this and was very important discovery. Here's the other two, the alpha secretase and the beta secretase. The alpha secretase cuts down lower, right, makes this fragment with about 28 or so amino acids, 26 amino acids, whatever, 26, I guess. And this guy cuts up here so that you can shoot a fragment that's 40 amino acids, right? You can see that when this guy cut and the gamma secretase cut, then you get this peptide fragment that's 26 amino acids. If this guy cuts, the beta secretase cuts, and then the gamma secretase cuts, you get this guy that's 1 to 42 or 1 to 40 amino acids. Kind of great. I keep saying 1 to 40, 1 to 42. And the reason is this guy here, this gamma secretase, this presenilin, is a little bit on the promiscuous side. It will cut a little bit on different sides, right? So just like this guy showed, it can cut here, like anywhere here, and give you slightly different sizes. It'll actually cut here and give you 1 to 39, so give a 1 to 39 fragment too. Okay. And this diagram just shows you the same thing. Here's the whole protein. There's that protease that I told you about. There's actually also this OX2 is a region that actually senses oxygen. So this is an oxygen sensor. I'm not sure exactly what that has to do, but it looks like oxygen sensors on the cut with the alpha and the gamma secretase, you get this fragment here, which is 26 amino acid, that's called P3. And if you cut with the beta secretase and the gamma secretase, you get this fragment here, it's either the 40 or 42 amino acid piece. This piece that's left in the membrane is called P7. It's sort of left attached there. This one, when you cleave this part here, right, your cells can get rid of that pretty readily, and this one they can't. I'll show you that in a minute. So the difference, let me tell you the difference here. I don't know that I put anything in here, but let me tell you the difference. If you look at someone who has Alzheimer's disease and they have plaque, remember that picture of plaque? There's a peptide in there, the beta amyloid from 1 to 42. So it's the larger one of these, 1 to 42. And the 1 to 42, that peptide, right? What's sort of what's one of the properties of that peptide? What's one of the chemical properties of that peptide? It has a very hydrophobic region because this part popped out of the membrane, right? And it's about halfway through, so it has somewhere, it has a dozen or so amino acids that are extremely hydrophobic. And so this peptide, which is small, really doesn't have much of a way to bury that. So that what happens is the hydrophobic regions interact with other hydrophobic regions, with other hydrophobic regions, and what do? Aggregate. We talked about that. Okay? Now, this is something with Alzheimer's. You and I, who also process this, I said we process this protein. We have our gamma secretase, or our gamma secretase cuts it away so that the fragment is 1 to 40, is 1 to 40. Right? So yours and mine cuts here. Someone with Alzheimer's cuts there. The only difference between the two are these two extra hydrophobic isoleucine and alanine residues that are there. If you take 1 to 40 and 1 to 42 in a test tube, we can make them and ask them to aggregate. This guy here aggregates 10 to the 5th times faster than this guy here. So the differences in two fragments is that the 1 to 42 aggregates much, much faster. And the difference between you and someone with Alzheimer's disease is that there's been a mutation that tends to cut more at the 42 than at the 40. If you look at our brains or in our spinal cord, we have those amino acids, those different things we do. But 1 to 40, the ratio of 1 to 40 and 1 to 42 in our brain or spinal cord is 9 to 1. But if you look at someone who has Alzheimer's disease, the ratio is 1 to 9. Exactly the opposite. So they have 10 times as much of this peptide here. It aggregates about 10 to the 5th times faster than what's in our brain. So they're aggregating and forming plaques faster than we are. That's the difference. Okay? Another thing I didn't put a slide in there for, but this gene, the gene that codes for this protein, right, occurs on chromosome 21. That have any significance to anybody? Go ahead. Trisomy 21, which is in Down syndrome. So someone who has Down syndrome, among other things, right, because it's the whole chromosome that's messed up. Everything on that 21st chromosome, they have an extra copy of that gene, so there's a lot of stuff that's messed up. None of the facial features or none of the observable things have anything to do with this peptide. It's other genes. But there's 3 copies of the gene that makes this peptide. So because of that, what happens is that there's sort of this imbalance, right, between the gamma secretase. There's not enough of the alpha secretase to cut all and make the 1 to 40. So then you get this beta secretase becomes more active and starts making more 1 to 42. Yes? So someone with Down syndrome has plaques. The plaques look identical to those in Alzheimer's disease. The difference is in Alzheimer's disease, there's a mutation in these processing enzymes. And in Down syndrome, there's an increase in the amount of the AP protein that AT processing that produces more of this 1 to 42 guy. Make sense? What else can I tell you? I'll think of it as I go. So So they're sort of interesting. I told you that this is sort of an unusual enzyme, this gamma secretase, the phenol-1. But it turns out that there are other proteins that are cleaved inside of the membrane like this. And this is a picture of a signaling system in Drosophila. It's called the notch. What is it called? It's called the notch, delta notch? I think it's called the delta notch signaling system. And delta is a protein that crosses the membrane in one cell, and notch is a protein in another. It looks like two proteins. It's actually just as it's dissolved notch protein. As it's processed, it's held together. It's kind of like insulin, right? The protein in the dimer is held together with disulfide bonds right here. So this is notch. And what happens is when two cells, and the delta protein has a binding site to the notch protein. So the notch and the delta. So when two cells meet together and their deltas and their notches come together like that, it activates the protease. And the protease that's activated, this guy's called TACE. And TACE is similar to, let me show you here, the alpha secretase is called TACE, right? So this is the TACE secretase. So what happens same in chocolate, the equivalent of the TACE secretase and precentillin secretase cut and cleave this protein in these two places. You do know that in the case of Drosophila, this is where we sort of get the idea that this part of the notch protein down there actually, after it's cleaved, goes to the nuclease and acts as a transcription factor. So it is a transcription factor in Drosophila. You know that. This molecule here, we think is some sort of can further signal in other cells. Okay? But what this thing here seems to do, right, is that when this is released, this is found in, what it does when this thing is released, notch causes adjacent cells to differentiate differently. So these are differentiating cells. Let's see, the liver. And they interact together. Right? Liver interact together. They release this fragment, this notch fragment. This notch goes to other non-differentiating cells and causes them not to differentiate in the liver cells. So this is involved in Drosophila, which is sort of the sizing of organelles, to keep the organelles from getting too big by developing differentiating cells in that organelle. So, okay, it is involved. Oh, and this, I'm sorry, I said Drosophila, this is in C. elegans, so this is in that worm. But anyway, this is very important. It causes other cells from differentiating. It's very important in the nervous system. Because what happens is that in development, right, as neural cells divide or arise or differentiate, they send out axons. And those axons make contact with a particular cell type, right? So the axon that innervates the muscle grows out to the muscle. But you never have several, there's only so many contacts between a nerve cell or axons and the target. You can't overdo it. If extra axons grow, they actually degenerate. They go away. Those nerve cells die. So there's a limited number. And one of the rules is like this shows here, is when these axons grow up and innervate a cell, right, then the notch and the delta interact. They release this fragment here of notch, which causes other neurons to differentiate into something else, astrocytes or whatever. So other precursor cells